Hello everyone and welcome to today's live broadcast. Primary motor and sensory cortical areas communicate via spatiotemporally coordinated networks at multiple frequencies. Presented by Dr. Fritzie Arce-Mcshane. I'm Susie Valdez of LabRoots and I will be your moderator for today's event. We are excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. Here's how this presentation will work today. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we'll make sure to follow up with you by email. You can also enlarge the slide window by clicking on the screen icon on the lower right-hand corner of your slide window. If you cannot hear or see this presentation properly, let us know by clicking on the support button at the top right or the Q&A button at the lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your web page and follow the processing of obtaining your credits. I would now like to introduce today's speaker, Dr. Fritzie Arce-Mcshane. She's a research assistant professor at the Department of Orgasmal Biology and Anatomy at University of Chicago. Dr. Arce-Mcshane's bachelor's degree was in physical therapy, her MA degree in motor learning, and her PhD in neuroscience. Her primary research interest lies in elucidating the neuronal mechanisms that underlie sensorimotor control and learning by using electrophysiological recordings from the sensorimotor cortex of behaving non-human primates. She has investigated the sensorimotor control of reaching, feeding, and the coordination of arm movements and posture. Her special interest lies in the understanding of the functional connectivity between the motor and sensory areas of the cortex that underlies sensorimotor integration. I will now turn over the presentation to Dr. Arce-Mcshane. Welcome.
Thank you, Susie, for the introduction. Hello, everybody. Thank you for attending this webinar on the primary motor and sensory cortical areas communicate via spatiotemporally coordinate networks at multiple frequencies. I hope you enjoy this segment. Sensory-motor integration is important and necessary in learning and performing motor skills. From the simple task of feeding, which we do every day, to driving a car, playing tennis, or even piano. All of these activities require interaction between sensory and motor processes that take place in our brain. Let's have a closer look at it using this simple diagram. So we start with a task goal. For example, hitting a baseball. And there has to be a change in our body configuration or in the environment in order to achieve the goal that we have at hand. So for this example, we have hitting the baseball. The motor cortex, which is involved in the production of our movements, will generate motor commands to our muscles in order to position our arm in a proper way to hit the ball. And then this movement will cause a change in our body and also in the ball that we're trying to hit. And there's going to be a sensory feedback as a consequence. So this could be like how fast we hit the ball, or like the strength of our grip, the position of the joints relative to each other. So all of this information will be sent to the somatosensory cortex. Now there has to be an exchange of information between the motor cortex and the somatosensory cortex in order for us to generate efficient movements. This exchange of information is kind of like a communication between these two cortical areas. And we know that neurons do not function in isolation. They form assemblies and networks. And the outputs of neurons have to be coordinated in time, kind of like what we call synchronization. So neuroscientists study interaction between cortical areas in many ways, and one of these ways is coherence. Coherence is the cross-correlation between two signals in the frequency domain. So for example, between spike trains of two neurons, or it could be spikes from one neuron and local field potentials from another electrode that we're recording from. And coherence has been suggested to represent like the coordination of the timing of synaptic inputs to a target neuron. So here, for example, on this slide, we see the inputs from one neuron. Okay, so here we have the action potentials and the inputs from another neuron. So with timing, like when you time the inputs from neuron A and neuron B, and they arrive at the target neuron at the same time, this will induce an action potential in the target neuron. Another role that coherence has is in supporting neurocommunication between areas. So one area, region 1, may be responsible for a particular attribute, for example, like the color of a car, and region 2 may be responsible for another attribute, maybe the speed of movement. So combining these two attributes, we come up with a concept of a fast-moving red car. Coherence will also have a role in the spike timing dependent plasticity and can actually induce long-term potentiation or long-term depression. So we see that there are many physiological functions for coherence. So we ventured into trying to understand the interactions between these two cortical areas, the motor cortex and the somatosensory cortex, during the performance of a motor task. So what we did was we implanted microelectrode arrays. So what you see here, okay, so it has like 96 electrodes for this array. We implanted them in the brain, in the motor cortex and the somatosensory cortex of 2 monkeys. And we trained the monkeys to do a behavioral task, which is to generate a tongue protrusive force. So how we trained them to do is to protrude their tongue and apply a force on a transducer right here, situated in front of their tongue. And they have to generate a force at the level cued by the target. So this is the target over here. So they need to bring the cursor that they see over here to the target, and that will be the amount of force that they're supposed to generate. So we studied the coherence between the primary somatosensory and primary motor cortex using the spikes and the local field potentials. So we did the spike-spike coherence, so meaning the spikes from the motor cortex and its coherence with the spikes from the somatosensory cortex. We also looked at the coherence between the spikes from the motor cortex and the local field potentials from the somatosensory cortex. And we also looked at the spikes from somatosensory cortex and the local field potentials from the motor cortex. So these measures of coherence represent kind of like for example, the spike-spike coherence will be an output relation of the outputs between the 2 cortical areas. And the spike field coherence will be a relation of the inputs from one area and the outputs in another area. So what we found is that the coherence between neurons and spikes and local field potentials from these 2 areas are actually dynamically modulated. So here I'm showing you the spike-spike coherence of a neuronal pair. This is a coherogram. On the y-axis, we have the frequency, which is 1 to 6 hertz. And on the x-axis, we have the time relative to the force onset. So this is the behavioral time. The color denotes the magnitude of the coherence, and you have here the scale of the coherence. So red is a higher coherence compared to the blue. So what we see here for this neuronal pair is that there is an increase in the coherence of this pair right around the force onset. And you don't see that in any other time relative to the force onset. Now we see another neuronal pair here, and we're looking now at a different frequency range, which is the gamma range, specifically between 30 to 50 hertz. And we see again an increase in the coherence in this pair around 400 to 200 milliseconds prior to force onset. Now we have seen this in many pairs of neurons. And in fact, we have here the mean coherence of all neuronal pairs. And I show here the coherence in the theta to alpha range, which is 2 to 6 hertz for the theta, 6 to 13 hertz for the alpha. And again, we see an increase in the coherence right around force onset. And this is the mean coherence across all neuronal pairs for the beta and gamma range. And here again is the scale, the range of frequency from 15 to 30 for beta, 30 to 50 for gamma. And again, we see here red spots, okay, around 400 milliseconds prior to force onset, and right about after force onset too. Now we see the same thing in the spike field coherence. Okay, so now remember that the spike field coherence is the relation between the spikes from one area and the local field potentials from another area. So the local field potentials in an aggregate activity of neurons, surrounding neurons, close to the electrode that we're looking at. And here I show the coherence, the spike field coherence for single pairs. So this is an example of MSF theta pair, so meaning that this is the spikes from motor cortex and the field from somatosensory cortex. And we see again that dynamic modulation. So it is task modulated. And we see an increase around between 200 to 400 milliseconds after force onset. Here we see the spike field coherence from a neuron from the somatosensory cortex and the local field potential from the motor cortex. And we see the same dynamic modulation. And here again are different examples of pairs of neurons in the gamma range showing the same dynamic modulation. What is interesting in this one, because we're looking at spike field coherence, is that the interactions between these two cortical areas are reciprocal. So there is communication coming from motor cortex towards the somatosensory cortex, and somatosensory cortex going to the motor cortex. And here again, we see the population of neuronal pairs of spike field coherence, okay, in the network of the MSF and SMF, in the theta range, which is the low frequency, and in the gamma range, which is the 30 to 50 hertz frequency. So we saw in those previous slides that coherence is modulated by the task. And now we see that this coherence is exhibited in multiple frequencies, okay? And there are differences though. Like in the theta band, which is your low frequency, we see a larger and stronger network compared to the alpha, beta, and gamma. Okay, so there is also coherence in these frequencies, but the networks are sparser and weaker. And we see that in the magnitude of the coherence here. And the same is true when we look at the spike field coherence. Now, again, this is the mean peak coherence, which is the magnitude of your coherence. And we see that in the theta, the coherence is much stronger compared to the alpha, beta, and gamma. What is interesting in this slide, though, is that we see a stronger theta coherence in the MSF network as opposed to the SMF network. While in the gamma coherence, it's the reverse. It's stronger in the SMF network as opposed to the MSF network. Now we look at the distributions of time of peak coherence. So we remember in those slides, we saw those red spots coming in at different times. Now we looked at the distribution of the time of peak coherence, and what we see is a pattern that is consistent across all networks. Okay, so this is the spike coherence. This is the spike field coherence between M1 neuron and somatosensory local field potentials. This is the spike field coherence between sensory neurons and local field potentials from the motor cortex. And all of these networks, we see a unimodal distribution of the time of peak coherence, which suggests that the theta coherence may be related to the generation of the tongue protrusive force. Now, when we look at the gamma coherence, the distribution of the time of peak coherence is actually bimodal. Okay, we see two peaks that are like around 500 plus or minus 500 millisecond relative to the force onset. And we see that again consistently across all networks, okay, the spike coherence and the spike field coherence. So this suggests to us that there are different processes that are mediated by the theta and gamma coherence. So gamma coherence may be suggestive of processes concerning sensory gating, attentional processes, or maybe memory encoding. So now let's try to sum up what we have been looking at. So we have seen the coherent activity between these two areas is modulated by the behavioral task. It also exhibits frequency-specific spatiotemporal properties, as we have seen that during first generation, theta coherence is prominent and exhibited by numerous paired signals. And for the gamma, for the alpha, beta, and gamma coherence, we see that this may be more related to sensory gating attention processes because we see them prior to or after fourth generation. And again, the coherent activity exhibits network-specific properties. So we see that there are differences in the magnitude of coherence in the spike field coherence between motor cortex and the somatosensory cortex. So now we have been looking at the magnitude of coherence, right? So another measure that we get when we do a coherence analysis is the phase of the coherence. The phase of the coherence tells us the lag, the time lag between the two signals. Okay, so here, and this is just a representation of how the different phase lags are, right? So here I show an in-phase relationship between the two signals. Okay, you see that the peak of this oscillation from one neuron, for example, or one local peak potential, coincides with the peak of the other signal. So we call that in phase because the phase is more or less around 0 degree. And then the antiphase, basically out of phase, when we see like the peak of the oscillation is in line with the trough of the oscillation. So there are antiphase. So the phase lag would be plus or minus around 180 degrees. And indeed, when we look at the distribution of the phases of neurons to all the local field potentials that they are coherent with, we saw neurons that showed a unimodal distribution of antiphase here, because this is plus or minus around 180 degrees. And you have an example of a neuron here, which is unimodal, but in phase with the local field potentials that is coherent with. There are also neurons that showed bimodal distribution of their coherent space, like this neuron here and another neuron here. So coherent space can give us an information in terms of the temporal organization of these networks. So what we looked at is to try to see what is the coherent space at the time of peak coherence. And so we plotted these polar plots. So this is basically a circular histogram of the distribution of the phase at peak coherence in the spike coherence and in these two spike field coherence. And we see that the distribution in the theta coherence of the phase at peak coherence is bimodal. Okay, we see that consistently across all these three networks. Okay, so it suggests that that in the theta coherence we have two subnetworks. So those paired signals that manifest a bimodal in-phase relation with their local field potentials or other neurons, or an antiphase relation with neurons and local field potentials. Now, we did not see this in the higher frequency bands, no? So like alpha, beta, and gamma. So in the spike coherence, the gamma distribution is uniform. In the spike field coherence, it is unimodal. So the bimodality is specific to the theta coherence. Now, so as I said, that the phase of peak theta coherence could suggest to us some temporal organization of the subnetworks. And indeed, when we compared the time of peak theta coherence for these two networks, the antiphase and the in-phase, we see differences across the networks. Okay, so in the spike-spike coherence, there is a difference between the in-phase and antiphase, and as well in the spike field coherence. And we know that here it even shows that the in-phase in the spike-spike coherence peaks first, right? So they have a time of peak coherence that occurs first. So now we can add the properties of this coherent activity between the two cortical areas, showing that there is a bimodal distribution of the phase at peak theta coherence, which suggests to us that there are two subnetworks that are involved, the in-phase and antiphase subnetworks. So now we go to kind of like a different theme, which is the spatial pattern in the motor cortex. So over the years, there have been growing interest in the spatial patterning in the activity of neurons in the motor cortex. And these are just like examples of different studies that look into such spatial patterning. And these studies suggest that the spatial patterning of activity in the motor cortex may relate to some physiological functions, like in terms of eliciting activation of specific groups of muscles, for example, the proximal versus the distal, or in terms of information transfer as well. So we were very curious to see whether we can see a spatial patterning of the coherence between these two areas. So we wanted to ask the question, does the spatial location of coherent neurons influence the timing of their peak coherence? And here we look at the spatial pattern in the orofacial sensorimotor cortex. I have here a representation of the array, the microelectrode array that I showed that we implanted in these two macaques. And they're snapshots of 100 millisecond activity. Okay. The orientation of the square corresponds to the location in the motor cortex and the sensory cortex. So here what I'm showing is the microelectrode array in the somatosensory cortex. So here, this is the evolution over time of the coherent activity. Each dot would represent the position of the neuron that we're interested in. So for example, so this is a 10 by 10 grid. So this neuron will be here, farther away from the central sulcus. So this will be the rostral part, and this will be the lateral part. So these neurons will be closer to the lateral border of the array. These neurons will be closer to the medial border of the array. The color of the dots represent the timing of their peak coherence. Okay, so we took at 100 millisecond slices of the coherent activity, and we see like some organization starts to happen 300 milliseconds prior to force onset. Okay, so we see that most of the activity is happening in the medial border of the array, and then progresses, okay, going laterally. So such that at 400 milliseconds after force onset, activity is exhibited by neurons located in the lateral border of the microelectrode array. So this represents to us some kind of a spatial gradient, okay, that is moving from medial to lateral. So now we looked at the mean spatial gradient of that coherent activity of these two monkeys. Okay, so here I show the microelectrode arrays we implanted from monkey Y and the microelectrode arrays that we implanted from monkey B. This represents the microelectrode arrays in the motor cortex, the green ones, and the lighter greens represent the microelectrode array implanted in the somatosensory cortex. And this is the central sulcus. This is the rostral part of the brain. This is the lateral part, the medial part, and the caudal part. So when we average the spatial gradient, so from each of the days that we recorded from these animals, we saw a pattern which is rostrolateral progression of coherent activity in the motor cortex, and that is happening here for the spike-spike coherence and for the spike field coherence. So this would be the MSF, meaning the spikes from M1, from the motor cortex, and the local field potentials from the somatosensory cortex. So we saw this in monkey Y, and we saw a more rostral progression for monkey B. It's just the same, it's going from the central sulcus rostrally. Now, when we look at the pattern, the spatial gradient in the somatosensory cortex, we see that the gradient of the spatial gradient is happening from medial border to the lateral border. Okay, so, and that's for monkey Y and also for monkey B. So it comes from the medial to the lateral. Okay. So what is the significance of the direction of these spatial gradients functionally? Right? So in a previous study that we have done, we looked at the activity of the motor cortex neurons and their relation to force. Okay? And this figure here shows a distribution. It's a bimodal distribution of the lags between the activity of the neurons and the force. Okay, so we see here a group of neuron that actually is leading force and another group of neuron that is actually lagging force. Okay, so now we try to look at the position of these neurons in the microelectrode array. Okay, is there some kind of spatial patterning of the position of these neurons based on their relation to force, whether they're leading force or they're lagging force? And here we see in blue the neurons that are lagging force, and in red the neurons that are leading force. This is the microelectrode array, so it's a 10 by 10 grid. This is the central sulcus right here. And we see that in this shaded green, light green area, we see more of the red dots. Okay, meaning like we see more of those neurons that are leading the force. So this suggests to us that neurons that lead the force are located closest to the central sulcus. Okay, and here you see more of the blue neurons, the neurons that are lagging force, farther away from the central sulcus. So we thought that this could explain the spatial gradient that we saw in the coherent activity of M1 neurons. Okay, when the spatial gradient moving rostrolaterally, right? So moving away, starting from the central sulcus and moving away from the central sulcus. Now, what could be the significance of the spatial gradient in the somatosensory cortex that we saw? So remember, the spatial gradient is moving from the medial border of the cortex, of the somatosensory cortex, and towards the lateral border. So previous studies have actually shown that the mechanoreceptive fields of the lips, okay, are located in the medial border of the somatosensory cortex and the tongue at the lateral border of the somatosensory cortex. Now, if we examine the tongue protrusion task, okay, so we start with an opening of the lips and then the tongue protruding in order to apply the force. So this can very well explain why we have that spatial gradient coming from the medial border to the lateral border. So now let's try to summarize and see what we have learned so far. We have seen that there are coherent networks between the motor cortex and the somatosensory cortex, and they involve multiple frequencies. Okay, so we see them in the theta, in the alpha, beta, and gamma bands. They're frequency and network specific because we see that that in the theta band, it's larger and stronger, and in the alpha, beta, and gamma, it's sparser and weaker. And the synchronous activity of the local and large-scale networks may be organized through multiple frequency bands for different cortical processes to operate at multiple temporal and spatial scales. So the fact that the coherence is happening at different frequency bands suggests to us that there are different cortical processes that have to be organized over time. And we saw that because we have the time of peak coherence in the theta coherence happening around the force onset, suggesting that theta coherence may be more related to force generation. And the fact that we saw the bimodal distribution of the time of peak coherence in the higher frequency bands, suggesting to us that the gamma coherence may be more related to processes involving sensory gating, attention, or memory encoding. And we also saw that there is a reciprocal interaction between the motor cortex and the somatosensory cortex. So what we saw in the beginning, right, that possibly that the motor cortex modulates sensory processing in the somatosensory cortex, and the somatosensory cortex transmits afferent information to the motor cortex for successful task performance. Okay, so because the motor cortex knows the kind of movement that it wants to generate, it also has an idea of the kind of sensory consequences that these movements will have. Okay? And in this way, it's able to transmit that information to the somatosensory cortex and prepare for that movement. And in the same way, with the information coming from the somatosensory cortex, the motor cortex can then adjust the commands that will come so that the movement will be more efficient and more successful. We also saw that there is information, of course, there is information in the coherence magnitude, and there's also information in the phase of the coherence. Okay, so the phase of the coherence reveals a temporal organization of subnetworks. And this is possibly because the brain wants to organize the activity of the neuronal ensembles at different time scales. Okay, so it has that flexibility to call upon different assemblies, okay, to be active at different times, depending on the particular function or the particular goal that the organism has. It could be also very helpful to segregate feed forward and feedback influences, okay, so that the behavior that we have, so we use different muscles and we use different motor patterns, and we want to have the ability and the flexibility to coordinate the activation of these different assemblies. And lastly, we saw that this coherent activity has a spatiotemporal pattern. Okay, so we saw that in the motor cortex, the neurons, the coherent activity of motor cortex neurons with other neurons and LFPs start from the sulcus and going rostrally. Okay, and in the somatosensory cortex, the coherent activity starts from the middle border, middle side, going laterally. And this pattern relates to the temporal relation of the neuron spiking to the tongue force. And in the somatosensory cortex, it relates to the spatial features of the neuron's receptive fields. And I'd like to thank people who helped me in this work. So definitely Professor Nicholas Hatsopoulos, Professor Callum Ross, and Kazutaka Takahashi, who are also the co-authors in this paper. Professor Yali Amit, who helped us with statistical analysis, and different people from the lab, the Hatsopoulos lab, for their help in the analysis and also discussion of the methods and animal care. Thank you very much.
Thank you so much for your presentation, Dr. Arce-Mcshane. We want to get right to the questions and input of the audience. So here's a reminder as to how the audience members can communicate with us. Questions can be submitted via the Q&A button at the lower left. If you are unable to get to your questions due to time constraints, Dr. Arce-Mcshane will be answering submitted questions by email. So let's begin with our first question. An audience member would like to know, there is a theoretical work showing the dependence of spike field coherence on the overall activity of neurons. Did you control for the differences in the firing rates?
A very interesting one, and indeed I actually have a slide to answer that question. So it's been a question that I've been asked about a lot of times. Oh, here. So it's true that there's actually a theoretical work suggesting the dependence of the spike field coherence on the overall neuronal firing, neuronal activity. Okay. And as a consequence, many of the studies have actually controlled for the firing rates of their neurons, trying to use similar firing rates when measuring the spike field coherence. Okay, so this is very good, and it's an issue that we should take care of. However, we know that in the motor cortex, the neurons in the motor cortex and the neurons in the somatosensory cortex have different firing rates. Okay, and it's understandable because they do different computations. Okay, so the motor cortex is to generate movement. The somatosensory cortex is to process those sensory information. Right? So we were hesitant in controlling for the rates of the firing of these neurons, but we wanted to address this issue of the dependence of spike field coherence on the differences on the spike rates. So what we did is to verify whether we could actually see this linear dependence. So what we did is, here is a graph where I plotted the rates, the spikes per second of neurons in the spike field coherence between motor neuron spikes and field potential from the somatosensory cortex, and the coherence. Okay, so here for this particular example, this is just one neuron and all the coherence it has with different local field potentials. Right? So here we see that there is no relation. Okay? There's no linear relation, and the same is true in the SMF. Okay, so what we see here is that in this particular experiment, we didn't see any relation of the mean firing rate of neurons to the spike field coherence. Okay, so we were very confident that that whatever differences in the magnitudes in the coherence that we see in these two networks is because of a real physiological difference, and not just because of the artifact of different firing rates.
Thank you so much. Another audience member would like to know, can the coherence between the motor somatosensory areas of the cortex be due to a common input in these areas?
Thank you. Very interesting question. Yes. It is possible that the interactions that we see between the motor cortex and the somatosensory cortex can be explained by a common input to these areas, right? So, so you have here the two areas, the motor cortex and the somatosensory cortex. We were looking at the interactions between these two, but this interaction may be coming from a third party right here, so going to the somatosensory cortex and going to the motor cortex at the same time. Okay, so and one possible brain area would be the thalamus. Okay, so the thalamus could definitely send simultaneous inputs both to the motor cortex and the somatosensory cortex. And indeed, there are many studies looking at thalamocortical connections. Alternatively, what we've shown you with the results could actually show direct cortico-cortical interactions between the motor cortex and the somatosensory cortex. And this is supported by studies showing that there are dense connections between the motor cortex and the somatosensory cortex. And that could be the substrate for the communication between the motor cortex and somatosensory cortex. Also because we know that these two areas, behaviorally, we need these two areas to communicate in order to efficiently produce a movement that is optimal and successful in achieving a task goal.
Here's a really great question. How critical is the coherence between these areas to the control of movement?
Yes, that is the big question, right? So, so we do see this interaction between these two different cortical areas, but how important is this interaction? So I would say that is very important, okay? But the data that I have shown you cannot actually answer to that question of its importance, of its vitality, of its necessity, okay? And we would like to do more research, more experiment in order to answer this question, okay? And what I see is that one way of answering this question is when we can actually perturb the communication between these two areas when we're performing motor behavior, okay? So if indeed the coherence, the interaction between these two areas is vital and necessary for the generation of movement, then if we perturb that communication, we would see its effects in the behavior. So we won't be able to probably successfully perform a particular motor behavior, or maybe it may not be that, what do you call this? It may not be that extensive. It may just cause some small effects that don't really disturb the performance of movement. But we still have to try to answer that question with more experimentation.
There are so many wonderful questions coming in, but we only have time for one more. Dr. Arce-Mcshane, what do you think is the significance of the differences in the time of peak coherence across those frequencies?
Thank you, Susie. Again, that's a very interesting question. So we see that that coherence is exhibited in the multiple frequencies, right? And then we saw that there is a difference in the timing of peak coherence. Okay, so in the theta, it happens very close to force onset. In the higher frequency band, it happens like plus or minus 500 milliseconds relative to force onset. Okay? So the different timing of peak coherence may suggest that, may give the system actually that flexibility in order to process different cortical processes at multiple scales, right? At multiple scales, spatial scales, and temporal scales.
Wonderful. I would like to once again thank you so much, Dr. Arce-Mcshane, for your presentation. Do you have any final comments for our audience before we leave today?
Well, thank you, Susie. Thank you, Susie. I just want to thank everyone in the audience for your interest in listening to this webinar. And I'd like to thank LabRoots again for the invitation and allowing me to speak in the Neuroscience 2017 event. Thank you very much.
Thank you again, everyone. That was Dr. Fritzie Arce-Mcshane. I would also like to thank LabRoots for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through June 16th, 2017. Keep an eye out for an email from LabRoots alerting you when this webcast will be available for replay. We encourage you to forward that announcement to your colleagues who may have missed today's live event. Thank you for joining us, and we hope to see you next time. Goodbye. Have a great day.